Nowadays, DNA is used in many modern techniques such as pregnancy testing and genetic testing. Many people have a basic understanding of DNA, but today I want to show you how DNA works for the human body. DNA stands for deoxyribonucleic acid, and deoxyribose means lack of oxygen. Nucleic acids are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus atoms and are divided into DNA and RNA. The basic unit of nucleic acid is nucleotide, and a nucleotide molecule can be divided into three parts: phosphate, sugar, and base. DNA is the molecule that carries the genetic structure, which is related to the growth, developmental functions, and reproduction of the organism. Also, DNA is the main genetic material, and genes are closely related to your life. Look, your blood type, type A, type B, type O, type AB, your height, your hair, your face shape are all determined by genes. Many people wonder how living individuals are constructed from DNA, and how do amino acids arrange themselves into a perfect protein. It's thanks to the blueprint of DNA, whose primary function is to tell amino acids how to arrange themselves to form the perfect protein shape. And DNA works in the first place because it wants you to meet your perfect self. It's unromantic. Now, I'm gonna give you a brief explanation of DNA replication, transcription, and translation. The site of DNA replication is mainly in the nucleus. This is the model of DNA, a molecule with two strands that twist around each other in a double helix structure. The sequence of each strand consists of four bases: adenine, cytosine, guanine, and thymine. The two strands are complementary. When there's T on one strand, the other strand corresponds to A, and C always pairs G. The two strands are parallel to each other, which determines how the DNA strands are replicated. RNA is synthesized in the nucleus. Using one strand of DNA as a template, a process called transcription, which simply means turning DNA into RNA. The translation process in the simplest saying is the transformation from RNA to protein. Perhaps this is a bit complicated, but I will give you an example to help you better understand. As a simple example, all the genetic information of life can be considered as a book, which I will call the Book of Life. This book contains all the information about you, and what language is the Book of Life written in? It is DNA. This language is only for four letters: A, C, G, and T. These letters are combined to form a sentence. Then I will call this sentence a gene. The cell is like a small factory in a living organism, and the Book of Life is your product manual. The engineers in the factory cannot understand the words written in DNA, so we need to translate the genes into another language, which I call RNA. A protein can be considered as a product made by the Book of Life, which is composed of several parts, and these parts are amino acids. The engineers in the cell assemble the amino acid parts according to the order of the words in the Book of Life and process them to obtain the protein. Anyway, we are born with the genes we inherit from our parents. I used to think that our genes pretty much represented our destiny, but our lifestyle choices have a major impact on our gene expression and our lives. We can make lifestyle changes by abandoning those self-destructive bad habits. Many small miracles can occur and even be passed on to future generations. It's kind of like playing poker. You're given some cards and that's all you can do with it. But we have to realize that you can play the pokers in a variety of ways, and that's where the environment comes in. Have you ever heard of the Human Genome Project before? It's an international research effort to sequence the human genome and identify the genes and contents. We used to hear the phrase, the 21st century is the century of biological science. This saying is said to have spread because of the Human Genome Project. The Genome Project will have an indispensable role in the treatment of disease, and the final goal of this project is to determine the 3 billion basic chemical composition of the human genome called nucleotide, and thus uncover the interrelationship of tens of thousands of genes related to human life, sickness, and death. And I believe that shortly, scientists will reveal much more than with the genes themselves. Type 1 diabetes is known to be an incurable, lifelong disease. Have you ever thought about changing your genes as a way to reduce your chances of developing type 1 diabetes? Previously, researchers cured the mouse of type 1 diabetes using a pyramid-shaped DNA molecule called tetrahydrobappone nucleic acids, but there are still some problems. As medicine continues to evolve and progress, new options for the treatment of diabetes have emerged, such as gene repair therapy. the fact that more than 90% of diabetes is a complex polygenic disease. Therefore, gene therapy also has limitations. But don't worry, there's indeed a strong possibility that the cure for type 1 diabetes can be achieved by genetic modification. In my opinion, I agree with this DNA technique if there is a high and certain success rate because everyone wants to be healthy. But there are two sides to everything. Changing a gene is still very risky because a gene usually has multiple functions. If the entire gene is changed for a certain trait, the other functions of the gene will be lost as well. What's more, if the technology is not used properly, it can lead to negative effects. Therefore, there are many aspects that we as scientists need to consider, and this is not a small project. Thank you for watching this video.